Today class we are going to discuss about mutation. Mutation was first introduced by the scientist Pico de Vries. He is one of the important scientists regarding the mutation. In the year of 1901, he observed the phenotypic changes in the evening primrose plant. The evening primrose is the common name of Eunothera lamarcana. It is one of the important plant. So he carried the research on this plant. According to Pico de Vries, the mutation is an inheritable sudden change in the genetic material of an organism. So, what is genetic material? It's genetic material. It may be DNA or RNA. The genetic material, both DNA as well as RNA, which are involved in hereditary function. Any change in the sudden change, any change or the sudden change in the genetic material is called as mutation. This is one important 2 more questions. Define mutation. The term mutation was introduced by Hugo de Vries. He observed mutation in primrose plant called Eunothera lamarcana. He said, mutation is an inheritable sudden change in the genetic material of an organism. Up to this, you are tried for 2 more questions. Again, the mutations are broadly classified into 2 main types, namely chromosomal mutation and gene mutation. First type is called chromosomal mutation. A sudden change in the structure and number of chromosomes is called chromosomal mutation. The chromosomal mutations are 2 types: sudden change in the structure of chromosome or the numerical changes. Change is called structural change. The second change is called numerical change. Okay, the first one. What are the changes in the structure of chromosome occurs? First one, the structural change in the chromosomes usually occurs in the cell division. Normally, there are 2 types of cell division. You know that one is mitosis and meiosis. During this cell division, there are error in the sense what if it is a particular chromosome may be deleted, a particular chromosome, extra chromosome will be added, or the chromosome will be inverted around 180 degrees, and the chromosome segment may be translocated from one part of the chromosome to another. So, due to cell division, some errors may be happen. Due to the errors, the structural change will occur. The second type of chromosome change is called as chromosome change in the number. The changes in the number of chromosomes, first one is, it is called numerical changes. The numerical changes nothing but what the chromosome number will be altered. So this is also called as pleiady. So there are 2 types of pleiady, one is euploidy and aneuploidy. The first type is called euploidy. And what is euploidy? So euploidy is a condition in which the individual has more than the usual number of diploid set of chromosomes. So euploidy means true, pleiady means numerical change. So it is a true numerical change that is occurs in the plant body or any organisms. Okay, so normally all organism contains a diploid set of chromosome. In the diploid set of chromosome, more than the usual number of diploid. So, for example, addition to the diploid, one more haploid may be added. For example, this one triploid. What is triploid? Tri means 3. So 3n will be there. 3n means 3 haploid set. So normally it was 2n. By addition of single n, it forms 3n. Okay. So the triploid plants and animals are typically sterile nature. They cannot reproduce. If the 4 haploid sets of chromosomes are present in an individual, it's called as tetraploidy. So in tetraploidy, there is a lot of advantages we are having when the plants are said to be tetraploidy. Suppose the tetraploid plants or advantages are they often result in haploid plus haploid. In aneuploidy, the loss or gain of one or more chromosomes in a set. The set means what? In the diploid set. Only one the numerical changes occurs in one number increase our number is lost. So it is called as aneuploidy. For example, the first type of aneuploidy is monosomy. In monosomy, mono means single. So from the diploid set of chromosome number, one chromosome is lost. It's called monosomy, also called as 2n minus 1. Next type, trisomy. In trisomy, one chromosome is added. So 2n plus 1. For example, 46 chromosomes are there, means 46 plus 1, 47 chromosome will be there. That's called trisomy. Monosomy, 2n minus 1. 46 chromosomes are there in diploid side. 45 chromosomes will be there. For example, nullisomy. Nullisomy means 2. Okay, nulli means 0 actually. Here the 2n minus 2. That's called as nullisomy chromosome. For example, due to the aneuploidy changes, how the chromosome changes that occur in the human body, Due to the chromosomal changes, what are the diseases caused in the man? Okay, first one is a Down syndrome. This is one of the commonly known as aneuploidy condition. See the first, see the what are the characters of Down syndrome? This condition first identified by the doctor, the named as long-term Down. The scientific name is long-term Down. In 1866, he observed the Down syndrome in a particular person called Down syndrome. Syndrome means particular deficiency, any defect. Okay. So it is a genetic condition in which there are an extra copy of chromosomes. For example, the trisomy. Trisomy in the sense 1 to n plus 1. It's called trisomy. So out of 46 chromosome, 21st chromosome, that means in chromosome number 21, the total 46 chromosomes are there. In the 21st chromosome, extra chromosome 1 is added. It is called trisomy. It will be asked in one more question. So it is associated with mental retardation, delayed developmental behavioral problems, weak muscle tone, vision, hearing disability. These are the common conditions which are seen in these children. Okay, called Down syndrome. It is due to aneuploidy. And the next type of mutation is called gene mutation or point mutation. So what is gene? See, part of segment of genome is called as a gene. Point mutation is nothing but in the several nucleotide changes, the millions of DNA is made up of millions of nucleotides. Any change in the base pair, any change in the base pair of DNA, suppose we take the nucleotide, in the particular nucleotide is so nucleotide is made up of one nitrogen base pair, one pentose pair, one phosphate. So the changes occurs in the nucleotide level is called as a gene mutation or point mutation. It involves substitution, deletion, insertion, and inversion. So due to this substitution, deletion, insertion, and inversion, a single or more than one nitrogen base pair is added or deleted. So gene alteration result in abnormal protein formation in an organism. It is called as gene mutation, or it is called as a point mutation. In this class, we have discussed about mutation and their types. Okay, this is one important 5 mark question. If you have any doubt, you can ask me. Thank you all.